Okay, we're going to pick up with part 2 now, talking about hemostasis. And hemostasis is just the arresting of bleeding. We're just stopping the excess flow of blood to an area where there's a wound. And it comes in 3 steps. First is vascular spasm. Second one is the formation of platelet plugs. And the last one is coagulation. We will go through each in turn. So first, vascular spasm. This is just when there is a constriction of a blood vessel. We're just tightening down, making the diameter of the blood vessel more narrow so less blood cells, and thus any other fluids, can get through. Ultimately, this can happen in two different ways. First is that the nervous system can act on the blood vessel, sending signals to the blood vessel, allowing it to constrict. Or we can have chemicals get released from either damaged blood cells or platelets, such as endothelin, which is a type of amino acid, or thromboxanes. And this is a class of lipids. Both work just like the nervous system reflex. They allow the blood vessel to tighten and constrict. Next step in this process is called platelet plug formation. And this is just when we have a bunch of platelets gathered together to prevent the flow of more blood. And the way it works is that we have a couple platelets that bind to collagen fibers on the blood vessel through what's called a von Willebrand factor, right here, along the collagen fibers. After a couple of those platelets bind, those platelets release different signals, things like your thromboxane or your ATP. And those signal additional platelets to come and bind to the area. Ultimately, you have what's called platelet aggregation that occur, which you have a bunch of platelets adhere to one another, as well as to the substance fibrinogen, which helps stitch them all together. And ultimately, when you have this complex of platelets and fibrinogen holding things together, you have what's called the platelet plug formed. And the last step for hemostasis is coagulation. And this is just the formation of the actual blood clot. A blood clot is basically just a mesh of all the different stuff we have there. So all the trapped blood cells, all the platelets from the platelet plug, any excess fluid, any fibrin holding things together. Additionally, you have what are called clotting factors. And these are just proteins that help control the bleeding. There's a bunch of different clotting factors, but there's really just one I want you to know for the coagulation process. So speaking of that coagulation process with forming clot, we just talked about the formation of the platelet plug. That's what I'm circling right here. But the actual formation of the clot is this process down here. So to keep this really simple, the way it works is that you have an enzyme. Anything that ends in ase is always an enzyme, called prothrombinase. And this is either formed through what's called an extrinsic pathway or an intrinsic pathway. So an extrinsic pathway just means that there are chemicals coming from outside the blood vessel stimulating the prothrombinase to be produced. Or you can have what's called an intrinsic pathway, which is where there are chemicals inside the blood vessel that are contributing to prothrombinase being created. Either way, we have either an extrinsic pathway or an intrinsic pathway, which ultimately lead to the formation of prothrombinase. That's what I'm circling right here, number 4. So from there, once we have prothrombinase created, ultimately that will act as a catalyst to convert prothrombin into thrombin. And the idea is that once prothrombinase converts prothrombin into thrombin, thrombin has two special roles. The first is that thrombin goes on to convert fibrinogen, which is one of our blood proteins in the plasma, into fibrin, which ultimately contributes to the structural integrity of the clot. Additionally, the second thing thrombin does is that it goes on to activate factor 13, which helps to stabilize the blood clot overall. So in terms of coagulation factors, factor 13 is the really big one I want you to know. But in terms of breaking the process, you need to know that prothrombinase gets formed from either an extrinsic or intrinsic pathway. We have it convert prothrombin to thrombin. Thrombin then can do two things: convert that fibrinogen to fibrin, and then activate factor 13. And then both fibrin and factor 13 ultimately contribute to stabilizing and allowing structural integrity of the clot. So in terms of controlling the clot formation, coagulation, the formation of that blood clot, only occurs when we have enough of a coagulation factor built up. So we have a certain threshold that we have to meet. And the one factor I'm having you know is factor 13. There are a lot of other factors at play, but just know you have to have a certain concentration amount of the coagulation factors to actually have a clot to form and hold. And of course, we don't always need clots forming. We have a lot of time when we don't have open wounds, so we need our blood to keep flowing. And to prevent clots from just forming haphazardly, we have what are called anticoagulants. And these are just factors that prevent clots from forming. So maybe even if we had enough coagulation factors in a given area within the cardiovascular system, we have enough of these anticoagulants to prevent clots from forming unnecessarily. So then once the clot's formed and the body begins healing, whatever the wound may be, the clot will undergo what's called clot retraction, which is just, it starts to shrink as damage becomes repaired. And ultimately, when that clot needs to go away, we'll have it dissolve through what's called fibrinolysis. So lysis, the suffix just means destroying, so it means we're breaking up something. And since the clot is largely composed of the protein fibrin, we're just breaking down that fibrin. And ultimately, we have a substance called plasmin, which hydrolyzes the fibrin, breaks it down. Okay. So one of the last things we're going to talk about for this chapter is called blood grouping. And this is how we determine the actual blood type of an individual. And the blood type is going to be based on the antigens that we have on the surface of our red blood cells, not necessarily our white blood cells or our platelets, just the red blood cells. And the idea is that based on whatever blood type you have a certain type of antigen that identifies it. With every antigen, you have what's called an antibody that can bind to the antigen on the red blood cell surface. When the appropriate antibody binds to the appropriate antigen, you have what's called agglutination, which is the sort of clumping of the blood cells. And ultimately, you can have also what's called hemolysis, which is where the blood cells get destroyed. So hemo, remember, hemoglobin, and then lysis is destroying. And there are two groups for classifying blood. We have the ABO groups, and we have the RH groups. And the one you're probably most familiar with is the ABO groups. So like I mentioned, based on whatever the surface antigen of the red blood cell is going to be what type of blood we have. So if you have just the A antigen on your red blood cells, you are type A. If you have just the B antigen, you are type B. If you have both the A and B antigens, you are type AB blood. And if you have neither the A antigen nor the B antigen, you are type O blood. And this impacts also what antibodies you have flowing around in your blood. So if you have type A blood or type A antigens, you're going to have B antibodies. You will not have A antibodies, because if you did have A antibodies with type A blood, your own antibodies would bind to your A antigens and destroy your own blood cells. So you're not going to have, your own body is not going to produce antibodies for its own blood cells. Otherwise, it would destroy itself. So moving on with antigen B, your B antigens are going to have A antibodies. Then if you have A and B antigens, you're going to have neither A nor B antibodies. And if you have type O blood with A nor B, meaning you don't have any of those antigens on the surface of the red blood cells, your body's actually going to have both A and B antibodies. So why does this actually matter? It matters because, like I said, you want to make sure that the antibody of your own blood doesn't match the antibody of your own antigens. Otherwise, your body will attack its own red blood cells. So if we have type A blood and we have the B antibody, there's a mismatch. So the B antibody does not match the A antigen, so we will have no agglutination reaction. But if we do have a match, we have type A blood here with the A antigen and the A antibody, we will have a match here with the antibody matching the antigen, and we'll thus have an agglutination reaction where there'll be clumping and hemolysis, or destroying of the red blood cells. So this matters with blood transfusion because we want to make sure that when a donor gives the blood to the recipient, that their blood types match, because if they don't match, you could actually get the patient very sick. If, for example, you had type A blood of the recipient, and then we gave the recipient type B blood, my mouse is weird. We gave the recipient type B blood. Type B blood has anti-A antibodies, so those antibodies are going to attack the type A blood of the recipient, getting them very sick. So we want to make sure when we do a transfusion, we move that blood from donor to recipient. We have a matching blood type, or a blood type that isn't going to cause an agglutination reaction. So on the right over here, I'm circling, oh, there we go. I'm circling a little diagram I made for you. It helps you identify what blood types are compatible from donor to recipient. So if you have the type O blood, you can give to type A, to type AB, and to type B. If you have type A blood, you can give to type AB. If you have type B blood, you can give to type AB also. And if you have type AB blood, you can't give to any of the three, but you can receive blood from all three. So if you have type O blood, you're a universal donor. If you're a type AB blood, you're a universal receiver. So ABO group is the first way we classify blood, and the second way that we classify blood is with the RH blood group. So there's two types. There's RH positive and RH negative. So if you are RH positive, you have a special RH antigen on the surface of the red blood cells. If you are RH negative, you don't have any of these antigens present. So most of you are probably familiar with hearing the positive and negative when talking about blood types. If someone has, or someone is A positive blood, that means they have the type A antigens, but they also have the RH antigens. Or if they're O negative, that means they don't have the AB antigens, nor do they have the RH antigen. So that's how that factors in with the overall blood classification. And a really practical example in terms of medicine is with the hemolytic disease of the newborn, and this has to do with our RH groups. So during a woman's first pregnancy, if the mom has RH positive blood, meaning that she has the RH antigen, and the fetus also has RH positive blood, some of the blood potentially, if there's a rupture in the placental tissue, can get across the placenta into the mom's blood. This can be unfortunate because you may have the production of RH antibodies with the mom's side, and the mom is now very sensitized to that antigen by producing the anti-RH antibodies. This doesn't really come into play, this bodily response, until the second pregnancy. So what happens is, if there is a subsequent pregnancy, and the mom then has those, excuse me, has those antibodies produced, with a subsequent pregnancy, some of that, some of the antibodies from the mom's blood may then come over to the fetal blood and start attacking the red blood cells through agglutination reactions and hemolysis of the unborn child. This can lead to a lot of complications in terms of developmental issues and sickness and so on and so forth. Okay, so the last thing we're going to talk about is diagnostic blood tools for this chapter. So a couple of quick things throughout. Blood typing, like we just talked about, is determining an individual's ABO group as well as their RH blood type. Next, we have what's called a cross match, and this is where we mix a donor's blood with a recipient's blood to see how they react, to make sure that the type A blood is mixing with the type A blood of the recipient, or whatever blood types we're comparing or mixing together. And then lastly, we can do what's called a complete blood count, which is where we basically do a complete workup of an individual's blood. So we can do the entire red blood count, and that would be a count per microliter of blood. We don't have a count of the whole individual's body. We have it as a sample. We can look at how much hemoglobin they have in that microliter of blood. We can look at the hematocrit, so what percent of the blood sample is red blood cells versus white blood cells or platelets. And then we can do a white blood cell count per microliter of blood. So that's all we have for chapter 19. I will see you all for chapter 20.